In the next hour, we're going to take you through Arise. And I know some of you have been here for the 4th year in a row. And we started 4 years ago with a dream. And I remember saying to each other, should we really do a day like this? There's nothing that we actually have yet. It's all fairy tales and rainbows. And now, of course, we got to show that we've actually accomplished something. So it's equally scary to do something a day like this when you have nothing to show for yet, but also after 4 years and no, it's not finished. And it will probably not be finished in 10 years. But let's at least have a look at what we managed to do together in those 4 years. One of the biggest points is that we first need to understand exactly how we can recognize a species. So an achievement is already building such a reference database. Do we have all the specimens? Do we have a DNA barcode of them? Do we have an image so we can recognize them so that we can build the AI? And do those different systems actually talk to each other through a common taxonomy? Tricky question. We've worked with one Arise team, but you will see from today we're actually going beyond Arise. It's no longer just Arise or just the 4 partners within Arise, but we're growing, also connecting to other programs. So by now we can actually really identify 2 big streams of work. And they're still collaborating because they're still joined in the same data management systems to make sure that whatever is produced or recognized on the DNA side, from sample to DNA, or on the sensor side, all the way to an AI recognition, that you can harness that data together. And that's the power, because as a researcher, as a policymaker, as a field manager, we understand that you want to know exactly what's there, and there's probably not one technology that will tell you everything. So you need to combine those technologies and even combine that with human observations. These are our phenomenon team leads. They have been really driving what you will see today. And they will take turns, and they will talk about all of their achievements. And I'm starting with Lotte. Yeah, that's right. That's right.
Okay. So, hi. I'm Lotte. I'm a team lead of the sampling team. And so we're building this DNA reference library, which will contain DNA of all species, multicellular species of the Netherlands. So as a sampling team, we are in charge of collecting all those specimens of the species. So let's see what we achieved the last 4 years and how we actually get from a sample to actual DNA barcode. So from collecting in the field to preparing and sequencing in the lab, storing a lot of data and analyzing that. And with that, we can update our target list and publish the results in our new DNA dashboard. So to start with collecting, we work together with a lot of species experts and organizations that actively collect for us. So they are the ones that bring in the specimens and the metadata. But of course, we need to identify and communicate to them which species are still missing. So that is where our target list comes in. And this is a freely open tool on the internet that everyone can use. And you can search on any taxonomic level. And within the tool, we clearly mark each species as still wanted, yes, we want one, or unwanted, no, we've got enough. So now you know what we still need. You need some collecting materials, of course. So we created a very standardized method for this. For the physical samples, we created different kinds of sampling kits. And for the digital metadata that we also need, we created a sampling app. So within this app, you can scan a unique code. And that code links the specimen to the metadata. And also within this app, again, we mark each species as wanted or unwanted. So that's there also, and that corresponds to the target list. And we are using this very standardized method because, you will see later, we handle a lot of specimens and a lot of data. And in the beginning, the first year of Arise, we actually processed handwritten field notes and all kinds of different Excel formats. But this simply doesn't work anymore. We need automation. And in the end, when the entire infrastructure is ready, this is also the way how we can get the results there faster to the collectors, to the researchers. So when we, as a team, receive the specimens and the metadata, we prepare everything for the actual sequencing in the lab. So this means pulling out the leg of a specimen or taking a bit of tissue, for instance, of a snail. And then we also take images of every specimen. So that serves as a kind of a digital voucher. And last year I already showed you this video. This is our Keyence microscopic camera. And that takes images of these plates that we use in the lab, 96 images in about 20 minutes. And they are stacked images. That means that it's really all in focus. The whole specimen is in focus. And this takes, for one plate, takes about 20 minutes. Before that took us like a day. So this is really also optimizing things in the lab. And then this is a new system which does actually kind of the same, but then for those classic museum drawers. So those are mostly insects, dried and pinned in a drawer, and it takes images of every specimen fully automatically. And this we can almost start using now, so we are very much looking forward to that. And we built this within Naturalis ourselves. So besides identified specimens that the experts bring in, we also do some larger insect trap projects. So this year we did one in Leenderbos in Brabant, together with Staatsbosbeheer. And this is partly to test our workflow. So can we actually start monitoring biodiversity based on DNA? And also to fill the target list, and then especially with the unknown species. So this resulted in more than 100,000 specimens already, and we not even processed everything yet. We are now in the process of sequencing those. So this is kind of a gold mine because this will contain for sure new species for our target list, but also for the Netherlands probably, and probably even for science. So this is really a gold mine, but a very big one. So now the next challenge is how to handle that. And if you have a brilliant idea maybe, come and talk to me, because I would very much like to hear it. So, some numbers. Since last ORISE Day, we processed 109,000 specimens. And if we take a look further back, you see that that's really a lot. And if we continue like this this year, we will reach even a higher number. So now you understand why we have to optimize everything as much as possible. So if you are a species expert or you know one that is not yet involved with ORISE, please contact me or one of my colleagues, because this year we are still sampling and we are still filling the target list. And we need every help we can get, because this is a national challenge and we cannot do that without the help of every species expert out there. So now everything's ready for the next step, and that is the actual sequencing in the lab. So Kevin will go and update us on that.
Thank you. So my name is Kevin. As Lotte already introduced me, I'm head of the sequencing team, so basically focusing on all the lab work. And it's getting a little difficult to sort of keep track of what's been happening. So I figured, just to show what we've been doing in the last couple of years, let's go back to the beginning. Let's have some scary pictures. Some of you might know this when we were doing lab work 10 years ago. This was like commercial DNA extraction kits, Sanger sequencing that you had to manually edit. I see some people flinch. And manual pipetting, lots and lots and lots of manual pipetting. So when I see what kind of things we're doing now, when I think back of, like, for instance, my own internship during my master's, the number of specimens I did during my entire internship is less than we do in a single day here. So it's crazy how far we've gotten. So those three things I just showed you are the things we wanted to get rid of. So first of all, these extraction kits are really nice, but they're commercial kits, so they're quite expensive. So we've basically thrown them away and started from the ground, developing these new homemade extraction protocols without sacrificing on the quality of the DNA. So we now have some protocols for animal tissue, specific ones for museum specimens, but also plants and fungi, currently working on soil, and then water and air are next. We're lacking a bit behind on the specimens that were collected, that Lotte just showed, but you see we are really ramping up now, and that's because we are introducing more and more automation in the lab. So here's an overview of some of the equipment we've bought in the last couple of years. Some of them are pretty simple, some of them are pretty expensive, more expensive than my house. But these are the machines that help us take away all this manual work. So we have a machine soon that will open tubes. It sounds really silly, but when you get hundreds and thousands of, for instance, soil samples a week, someone needs to screw open all these tubes, add some buffer, close them again, shake them, open them again, take some buffer out. You can imagine that finding someone to open and close tubes it's a bit of a difficult thing nowadays because people don't want to do that anymore. So we have a machine for that soon, but also like for the DNA extractions and for a lot of the work in the lab. We have some cool machines now. Most of them look a bit scary, but they're actually helping us quite a lot. So just also to give some numbers here. So I took a look at what these machines do for us and how much time we can save using these machines. So in a situation where we would say we do 50,000 samples a year, we actually saved about 1,700 hours of manual work, which is more than a single person does in a year on a full-time contract. So that's actually really great, because then we open up these hours for the technicians to work on like exciting stuff like improving protocols instead of just pipetting plates all day long. Sanger sequencing, I know people still use it. It's a lot of work. So we've been looking into sort of replacing that technology. We've opted for nanopore, which we've been doing now for a couple of years, and it's actually quite great. And actually also this is a sequencing technology that keeps improving. It's getting cheaper and cheaper and cheaper every day. So this is sort of the three generations that exist now. We're currently looking at incorporating the third generation a bit more. So before we used to do sequencing of like, say, 2,000 samples overnight. Now we can actually do it in an hour. So you can see we're sort of ramping up the speed. And it's cheaper as well. This is really nice. So they once had a goal to sequence a specimen for less than €1. We're getting closer and closer if inflation doesn't bite us in the ass. The next step, so we've been working a lot on optimizing these workflows, doing more and more and more samples. But what we see now is we're also generating more and more waste. So our next goals is looking a bit more into the sustainability, because we are doing biodiversity research. We don't want to pollute the earth with more plastic waste. So this is one of the machines I showed earlier in a picture. This is called a tip washer. So what this machine does is it takes the pipette tips that we used, goes through a few wash cycles, and then we can reuse them without any contamination left. So we can actually save a lot on plastic waste and, of course, money as well, because these tip boxes are also getting ridiculously expensive. Of course, we're sequencing a lot of species that were given to us, but we're also looking into finding new species. So at the Westerdijk, there's a lot of work going on finding all these fungal species that live in the soils. So they've given me some numbers, and it's actually pretty impressive, because they've now already processed more than 800 soil samples from all over the Netherlands, except for Overijssel. I don't know if anyone is here from Overijssel. Please send some samples to the Westerdijk. Resulting in roughly 7,000 fungal strains, with a lot of species, and also species new. So there's a lot of new species being discovered here. Maybe some of these plates are familiar for the people that already saw them in our showcases or town halls. But a lot of these are being published on a almost weekly basis. So it's really great. Follow these for more, and hopefully we'll get a lot more species from this as well. Like Lotte just said, also from the Leenderbos, I expect to find some things that we haven't seen before. This is all about the lab work, but then of course once we get sequences rolling out, there needs to be some bioinformatics, and this is Rutger's department.
Yes, good morning. My name's Rutger, I can just follow up on what Kevin said. At some point, the technology push becomes incredible, right? We now have these machines which were just science fiction back when I first started doing a little bit of biology. And now we're also in a situation where you no longer like visually can edit the trace file of one Sanger sequence or whatever. We have machines that are just incredibly powerful, and we want to squeeze the most out of them. And then the first challenge that you actually have on the bioinformatics side is, okay, these machines can spit out loads of data, so much so that you don't throw in one specimen, highly wasteful, a whole bunch have to go in. What comes out on the other end is just little puzzle pieces, which don't normally say where they came from. So the first trick is to make sure that you can sort. If you do multiple specimens in, this is just 3, in reality it's way more. What comes out on the other end? How do we sort that? Well, here the trick is that the people in the lab attach a little bit of synthetic DNA to each sample, it goes in the machine, okay, then your bucket of data comes out, but because we know what we attach to each sample, we can now sort that and then have the data for each individual specimen in its own little data set. And this is, you know, of course we're doing a little bit of gatekeeping as experts, so we're calling this demultiplexing. It's just sorting the raw data. Okay, so we've demystified this part. A bunch of specimens go in, loads of data comes out, let's sort that and call it demultiplexing. So you see 3 specimens went in, 3 data sets come out. Scales to much larger numbers, but the principle is the same. Let's see if we go forward. Okay, so what comes out actually, and I don't want to torture you too much with this data, but what comes out is called FastQ. So now we've zoomed in on our one little ladybug, which is kind of going to be the star of the show in a couple of talks today. We've got a little ladybug with its own sorted data, and that's in what's called FastQ format, which is just a giant text file. And so on the text file, there's records that each span 4 lines. Okay, there's some IDs in there. There's the actual ACs, Ts, and Gs, as you can see. And then 2 lines below that, there's some quality indicators. So some symbols that indicate this little thing that went through the pore, through the science fiction system, was read with this much certainty. So the first step is to then see, okay, which of these observations were kind of bad, and can we clip those off? And this for hundreds, thousands, millions of these records, so this is all automated. Then we've done some initial QC on the data, and then we want to see, well, actually what's in the bucket for this particular specimen, because it's not really unambiguous. There's some substructure in these puzzle pieces, so the next step that we then do is some basic clustering. Because, well, there's the ladybug, there's maybe also something that lives on top of the ladybug or something inside it, or maybe there's a little bit of contamination in the lab or whatever. So we want to cluster, and what you see then on the right is kind of what that looks like. So here, a picture that every one of these little balls is one of those FastQ records. They're clustered by similarity, so you get these kind of islands. And for example, something like 3 of these separate islands for one specimen, that's kind of typical. Like that's kind of what you could have. And then, for example, one of them is the actual barcode gene that we're interested in, and the others could be maybe a contaminant or maybe a nuclear copy of the gene. There's this weird process where these barcode genes, and in general genes, kind of occur as broken copies elsewhere in the genome, and the lab process might accidentally have those as bycatch. And so we don't want that. We want to see what's what and then try to focus on what we actually want to have. So we've done the clustering, and then over each cluster you can compute a consensus, like the average sequence for that cluster. So then we have 3 of these average sequences. And hopefully one of them is the right one. One hopes. Okay, now let's focus on one of these consensus sequences. There's a bunch of different checks that we do to see if this might be the right one. And the first check that we do is what we call structural validation. So here's how that works. A gene, right, is actually a stretch of DNA along that rope ladder with actual, there's almost like Morse code that says, okay, start transmission here, and then there's the code that in this case actually translates to a protein, and then there's a kind of do-do-do stop. And so what we look for is whether that's intact in the consensus sequence, because if it's like a broken copy, then some part of this is kind of messed up. There might be an early stop, or it might translate to something really implausible. So we screen all these consensus sequences against a model of the gene and see, okay, are we close enough to that model? Then at least we are structurally valid. It's probably not a pseudo gene, it's a real gene, which could still be off target. Then here comes the fun part. So if you like Sudoku, this is going to be great. On the left, we have a part of the tree of life which is unknown to us, right? We know, of course, they're species. We know that they're produced by evolution. We know that there's some kind of tree shape. We don't know exactly what it is. And we might be missing stuff, obviously. In fact, we're missing more than we know, right? The actual diversity out there is enormous. In the middle is, let's say, the known knowns. So, for example, what we're looking at here is the global database for DNA barcodes, and for that same family, that system called BOLD might have, let's say, 4 species. Great, okay, so there's 4 species there, and then on the right, that's us. And we take BOLD as a kind of a point of departure. We've got our national checklist, and so we can cross-reference, and we can see, oh, this is great. The, I guess, bluish-looking dragonfly thing, which is in BOLD, occurs in the Netherlands. So now we have kind of like a proxy, an initial thing that we can use in our target list, which might be kind of an old record or not from this country. So it's kind of a placeholder, but it's a good start. The same for the bee-type thingy. Yay, we can carry that over as well. And we know from the checklist that this ladybug also occurs here, and it looks like this thing has been coming down the pipeline, right? It's been sampled, it's been sequenced, we have some data. Could it be that we now have a new record for this new ladybug? Okay, here's the tricky part. If it hasn't been sequenced yet, ever, there's no way to positively confirm that that sequence is of that species, right? The code doesn't say, hi, I'm a ladybug. It just says ACTG. Is it plausible that this would belong to that species? So we're going to go and check with the known knowns and see if it is kind of plausible, right? So here the idea is, well, you know, these barcode genes, they are passed on through, you know, lines of descent. So closely related species are recent inheritors from the same common ancestor, so their sequences must be similar. So what we then want to look at is, well, is our candidate sequence kind of plausibly fitting into this pool of genetic diversity at some higher taxonomic level? Right? We can't check species to species because it doesn't exist yet. We might check at maybe the genus level. That could also be very undersampled. Like really all these databases are very gappy, so maybe that's not even enough resolution, let's say. So we go typically to the family level, and then we can see with what we know of the genetic diversity in that family, possibly worldwide, doesn't have to be in this country, does this sequence kind of make sense in that wider picture? And so what's really essential here is that these specimens and these sequences that we're validating have some kind of initial identification attached to it, which we can then project into these databases. So we've got our ladybug, and it has to be annotated in such a way that we know, we'll get back to that. We have to know what it is with reference to the Dutch species register, so that we can then map that onto these global databases and see, okay, does this actually make sense? This is, okay, I'm kind of belaboring a point here, but we can only tell if something is valid if we know what it's supposed to be. Other than that, if it's from trapping, it's really great that we're collecting this data going forward if maybe other methods arise, as Lotte also mentioned. But for now, this is the best we can do. We need these annotations to at least check if this made sense as a sequence. So that's for the bioinformatics, and if it's all, kind of in order, then we have a sequence that looks plausible, and that can go into the larger system. And about that larger system, Chantal will tell us more.
Good morning, everyone. My name is Chantal Huibers. I was that spider in the middle of all the teams, and my team works on managing all this data. So, as you've just seen, we get a lot of data, and we have a whole bunch of different types of data. There's data about what kind of specimen has who sampled where at what time, specimen metadata, relatively simple. We have barcodes, we have all this tricky information that Rutger grabs out of his bioinformatics pipelines. We have images from the keyends, but as my next colleagues will tell, also from sensors like wildlife cameras and insect cameras. We have sounds, and then we have a whole bunch of AI results. And we put this all in, thanks to Jacob. This is, oh, wait, too fast. This is our little factory, a cloud full of biodiversity data. And we need this factory to actually manage all this data, to then bring it in a structured way to platforms such as the Arise platform, with different services for our end users. But we also want to bring this to other infrastructures, such as Bolt and GBIF. So there's a lot of things happening in this factory, and I can't show pretty images of robots in the lab, because it's just a screen full of code. But what happens here is still very important to maintain the quality of all this data. So we do a lot of validation. This is validation on whether the format is right, whether someone actually gave us a sample from 2027. No, it's not 2027 yet, so it can't be. Whether the coordinates are actually right. All these kind of checks that we do before we want to have it in the system. Because garbage in is garbage out, and we don't really want that. We want it to be authorized and quality of data. We also need to harmonize data from different sources. I'll give you an example of why we do that shortly. We manage all this data and we make it exposable to other systems. So for this first pipeline or circle, as we showed it, sample to sequence, at the moment we work with 3 different sources of data. We have ADA, that's our gold for metadata standards, but we do still have people that hand in sheets with data. Luckily, they're standardized these days. The sampling team has done a very good job in making sure that all the Excel sheets that come to us actually have the right and correct format, but still we want to validate that. And for now, we have all the sequence data. These are the raw sequences coming from the nanopore. We process this in what we call the DNA data domain within this big biodiversity data lake, the factory. And at the moment, our consumer of this data is the DNA dashboard. So I just said that one of the steps that we do is harmonize data. And one of the reasons why this is important is because we can get exactly the same data from different sources. For example, ADA and the sheets both give us information about what kind of sample this is. And for example, who collected this sample? Now, this is getting a bit tricky into data modeling, but from ADA, we actually get the who was this in separated across 3 fields. So Elaine is in the first name field, Om en Kloeke is in the last name field, and then Van is in some other field. Whereas in the sheets, there's just one column saying this is Elaine van Om en Kloeke. And Elaine only really uses Ada, but in case she would have handed in sheets, we actually want to make sure that in the DNA dashboard, she sees all her data. So in our DNA domain, this is one type of information, identified by. And we have the same for location, for all other kinds of data. So making sure that we can harmonize this across different sources is for us really important. We've been processing a lot of data. I kind of feel that sampling and then sequencing is lagging a little bit behind, and we're also lagging a little bit behind. But I checked yesterday. We have more than 1,000 ADA records. I don't know if that's kind of what you think we've been putting in. Actually, there's quite still a lot that don't come through our validation. So we need to have a chat with all these samplers or with the ADA system. But this is a good way to actually make sure that all the information that we need, so we have minimally required metadata, we can't do anything with a sample that doesn't have a latitude and longitude, because we want to be able to also send that to GBIF later, and they don't want just, well, there was somewhere in the Netherlands maybe a ladybug. We have more than 8,000 validated sheet records, so it's still quite a lot of sheet data. If you're a sampler, please start using ADA. We like that. And we have more than 120,000 DNA sequences already in that. And one of the next things is I actually match that with the bioinformatics pipeline from Rutger. All goes into the DNA domain. Oh yeah, this is what's coming next. So next steps is to actually also ingest the information from Rutger's pipelines. So we don't only have the ACTG sequence linked to each sample, but we also know whether it's structurally valid and what kind of identification came out of that. I'll show you the DNA dashboard in a little bit, but what it has at the moment is all the sample metadata and the sequence, but not yet the identification results. And once we have all of that in our DNA data domain, we can also start exporting that to Bolt. So we've come a long way from just starting to collect all this data to actually making sure that we can process it. The DNA dashboard was not developed by my team. It's actually Lotte's team who's been working hard on it, but otherwise we keep switching up here. So the DNA dashboard's live. This is where everyone who's handed in samples for a RISE can look up the samples and check their data. And if we have already a sequence for those samples, you can view that there as well. So now, of course, all of you think, I want that as well. Talk to Lotte in the break. This was the first circle, DNA samples, but we also do sensors. And I'm handing over to Emil, to start introducing that bit.
Thanks a lot, Chantal. So the monitoring and demonstration sites are places where our team tries to establish which sensors make sense to monitor what. So here we see an image of insect monitoring device, and the idea is to try different types of insect monitoring, for example, at these locations, before rolling this out over the entire country to monitor nationwide. See the innovation potential by these sensors, that's also our goal, as well as to connect to all sorts of users. And I will, before zooming in on one specific case, would like to show the types of devices that we have in the field and that we are evaluating on different locations. And at one end, you see the audio sensors. We have things like acoustic devices in the audible range, but also in the ultrasonic range of different brands that have different formats, as well as insect cameras. That's the pictures in the middle, that ranging from the one that you saw with a yellow screen up to a camera that specifically monitors nest boxes at the middle lower end, the insects that birds bring to their chicks. And at the right-hand side, sensors like radar, upward-looking radar, looking at bird biodiversity, coupled to cameras that can observe the same birds, and some environmental sensors like the water temperature sensor. And what I will highlight today are the wildlife cameras at the lower right, where you see the rabbit. Also highlighted perhaps in numbers at the very end, also in this part of our project, lots of data is being generated. And you can imagine that having this audio and these images requires a big effort to tag animal IDs to it, or IDs to everything that it's observed, but that's material for the next presenter. So we focus our efforts on different environments in the Netherlands, terrestrial environments. Hopefully we can expand it in the future years to also aquatic environments, perhaps in collaboration with other projects and parties. So these are the areas we're looking into now. And there's additional sites where we have not we're not running the operational stuff, but trying out individual sensors as well. Now I'd like to zoom in on the Amsterdamse waterleidingduinen, terrain managed by Waternet, and you'll later today also hear more presentation and also some demonstration workshop in the afternoon is also focusing on that. So the dune area is harboring species like this rabbit, the fox, and the fallow deer. And many of these are photos taken by the wildlife cameras, are observed there. And in fact, we have a beautiful website showing different story map that you can access here, which gives a bit of an integrated view of all the sorts of information that is collected at these locations. The map already shows how many devices are there, and you can imagine they can observe a lot of wildlife dynamics. A small illustration of what passes by is seen here. So variations in day and night, different kind of species, different kind of habitats. However, this information, and that's what I would like to highlight here, I'd like to zoom in on the rabbit, doesn't sit in isolation, and that's where biodiversity monitoring per se, focusing on what's occurring in the Netherlands, touches with the terrain managing organization, thinking about how can we make sure that biodiversity thrives, what is the trend in our terrain, in our current biodiversity status or species. And this is a result of terrain monitoring data by the Waternet people, monitoring rabbits on transects already for many, many years, ranging back from 1984 up till this year, seeing these big dips with two diseases occurring, very dramatic infestation over the past 10 years or so, and an autumn and a spring trend. And if you map this in space and other monitoring that takes place, pellet counts by these animals, you see also that spatially the rabbit population is kind of concentrating along the North Sea area. As a result of this declining trend, the population is managed by, or at least recovery is attempted by releasing healthy rabbits displaced from elsewhere at these locations with blue stars. Interestingly, to protect these rabbits from and have them getting accustomed to the terrain, they are released in fenced areas with the idea that they would stay in these fenced areas for a while and then move out into the wild. Our wildlife cameras have detected that they are good climbers. In fact, you see a little time series of a rabbit climbing a protective fence to go out into the dunes and discover the rest of the area. This is an illustration of what kind of behavior that we never anticipated is in fact seen in this wildlife. And to highlight some more, we also of course see diurnal patterns, activity patterns, space use at the right hand side of the map, interactions with other species, predators. In this case, we can have an idea about whether there's any avoidance happening or by being afraid in a landscape of fear of these predators, and perhaps competition between hare and rabbit, as it is supposed. Cause and consequence is not known in this case. All food for thought and very interesting material for further study. I'd like to end with this slide to emphasize that, this biodiversity collection information and the demonstration site are important steps in managing the terrain, getting to know the biodiversity in the Netherlands, but also to enhance it. And we hope with our partners in these different sites to put the picture together of how these demonstration sites work. And with that, I'd like to move on to Jacob Kammer's presentation.
Hey, good morning everyone. Bear with me, we're almost at lunch. A lot of information today. So my name is Jacob. I work at the University of Twente as a computer scientist. And my team is working on digital species identification. I try to keep this simple. In fact, by the end of this, I hope you're a bit underwhelmed, because what we're doing AI is quite complicated. But in fact, most of our users are not AI developers, but users of AI, right? So we try to make this as easy as possible to use it to scale up biodiversity monitoring. So we're trying to enable large-scale automatic recognition of species by using any type of camera or audio sensor. And in the next couple of minutes, I will show you an example of how you can use ARISE today to do automatic species recognition for any camera or audio sensor. So Emil already introduced this variety of sensors. They're all different types, all generate different types of data. And by the end, after using ARISE, we will provide you with organized data and identified species in this data. In general, this doesn't matter what type of sensor you're using, you're always placing it somewhere. So what you see here is an example of sensors in Utrecht. These are deployments of sensors. And they're usually grouped. It could be a habitat type or depending on your research question. So in this case, this is Emily's data. She's interested in comparing rural areas with urban areas. And as you can already see, this is called a deployment site in our system. So in general, when you use ARISE, we really try to help you structure your data. So this one-side structuring to help you organize this plethora of data. But it's also standardization, right? As Chantal already mentioned, we want to make this data interoperable with other platforms. So usually data is organized in projects and sites. Every site contains multiple deployments of sensors, and every deployment is at least a sensor, a location, and a time period. And then this list goes on downwards, because we have a lot of different types of sensors. So this is what it looks like. Yes, so I'll show you together. We'll add some data. So this is how you create a deployment in ARISE today. So I'll give it a name. We assign this deployment to a project. An actual site. This is data from my backyard. Of course, a sensor. This is my first wildlife camera, and I can use the map to show the location. So as you can see, I live in up north, and the deployment range of the sensor. Now, some sensors already send data automatically, so there's no need to manually upload data. But of course, there's now this is quite recent. It's also possible to upload data manually. So, for example, put an SD card. Select a folder and upload your items. Currently, our media browser, where you upload the data to, is handling more than 31 million media items steadily. So that's good news. And it's, of course, rapidly growing. Now, after you upload these images, you can browse them, you can filter them, and the next step is to create a data set. So in this case, I'll call it cats and birds in the backyard. And now we want to use AI, right? So the next step is to start an analysis run. This could even be automated, but right now you still have to press a button. So we hope it's not too much effort. So you select an algorithm, in this case TrapperAI. These are off-the-shelf algorithms. We didn't make these. These are available. They usually live in repositories, but now we try to incorporate them into ARISE so it's easier to use. So you select the algorithm, you select the data set. The job name is even automatically generated. Now here you can also, so I can press start analysis, and that's it. I can also select another algorithm, in this case DeepFawn, and also press start analysis. So this is the only action I've done so far. And this will run on the supercomputer, more on that later. So last year we had this list of algorithms that were readily available in ARISE. Now this list has grown over the last year. So you can see the media type column, there's both image algorithms there, but also audio. So the examples you see here are usually visual because it's nice to show you guys. But be aware that now you can also upload audio data and use, for example, the Aves Echo algorithm that's being developed here at Naturalis. And of course, the goal is to grow this list as we go on. So what do we do? This is, of course, ChatGPT, but we automate the deployment of algorithms to process all your data. So you can imagine like a single lane, in this case, a single lane highway to a factory that processes your data. And that works, but it was not the most fastest system. So last year it took about 7.5 hours to process 26,000 media items. The team has done great work over the last years to make a multi-lane highway. So data is processed more in parallel at the moment. So currently it takes about 15 hours to process roughly 700,000 media items. This is without optimizing the algorithm itself, but purely how we automate the analysis run on the supercomputer. While this is being processed, the dashboard is showing you information on the analysis run, what it's doing, and also it informs you in case something goes wrong. This whole part has also been optimized, so it's more robust. So if something goes wrong, it will automatically retry the analysis run until it really fails, and it will tell you. But usually it completes successfully. And when it does, it gives you back the results. So in this case, it's a visual algorithm. So the media browser, you can use it to browse the media and also overlay the results. So in this case, you see the trapper eyes correctly identified this bird as a bird, which is quite high level, right? It's not a species level yet. Deep Fawn has correctly identified my son as a human, and I was very confident about it. But you can also easily compare algorithms. So here on the right, I can select trapper eye, and there you can see trapper eye thought his boots were a dog. So this is already showing you the limitations of where we are now. More on that in a minute. So now we have run these analysis runs for multiple algorithms in this media browser of 31 million items. How do you make sense of it, right? So now we get back to structure. So you can use filters in the system to filter your data based on deployment, sensors, whatnot. But you can also use the results of the algorithm to look for birds, right? So in this case, it's high level, but it's already helping you a lot if you want to do something with birds. So the next step is to, you may want to identify them to a species level. And then this is really helpful. So like I said, algorithms make mistakes, and unfortunately a lot, right? Because what we do is really challenging. We want to use sensors in various environments, various conditions. So of course there are more and more algorithms out there. They're getting better and better, which is really good news. But we need a way to get algorithms to work for you and your project specifically. So there's obviously no human here, and that bird is not a dog. So what we need is, first of all, when you're looking at the images, intuitively you might want to just correct the algorithm, right? So that's not a bird, that's not a dog, it's a bird. So we want to provide that in the next year. But in general, we really want to help our users to annotate new training data, right? So in the case of the birds, we're now making a connection to annotation platforms. There's a lot of them out there. So it will be very easy to select all the birds and send them to a platform so you can easily identify them at a species level. And then, of course, those two combined together, you can use it to improve the algorithm. And this is, I think, helping a lot of users already at this moment. So when you do this, multiple people have similar data and annotate data in their own projects. But if we don't speak the same language, this may become more difficult to use each other's data, right? And there's a huge opportunity if we all use a system like Arise, that we also use each other's training data to improve the algorithms overall, which then benefits everyone again. But in order to do this, as a huge challenge, we need to speak the same language. And that's when we talk about ontology management, so that when you annotate more data, that it's easy to use common names that other people also use, right? And last but not least, all this data is synchronized with the BioCloud. So you can already see Chantal coming up, and she will highlight again why this is important and how this is done. Thank you.
Now it's really getting close to lunch. I won't keep you too long, but it's me again, because as we said, we've got two circles of data. We've got the sample to sequence, and we've got the sensor to AI data, and all of that is managed in this factory. Previously, I talked about how the sample metadata and all the DNA barcoding data goes into the DNA domain, so we also have a media domain. The in the same system. They're very closely related, but they have a different data model, so we can actually handle the data separately from each other, but also have them connected. At the moment, we mostly have Diopsis data in our media domain, over 40 million Diopsis images, which luckily are easily handled by the DSI portal that Jacob just showed. And soon we will also make sure that we add all that data that people can now upload themselves in the sensor portal. And together, when you then have DNA data as well as media data, you just saw the research that's going on, for example, in the Amsterdamse waterleidingduinen, we can really start combining this data. So if you have a malaise trap and analyze all the DNA sequences out of that malaise trap, but you also have a wildlife camera, or no, not a wildlife camera, the Diopsis camera next to that, or air eDNA, in the future we really make it easier to combine all that data and compare the results. What's next? Really getting all the digital species identification data, so not just the media, but also the results from the AI into the media domain. And then we can get derived occurrences that we can actually share with GBIF. So there's more to come. And then there's also the really not so sexy stuff. So managing access to data is really important. ARISE is building infrastructure that researchers can use while doing research. So a lot of them are still in the process of publishing their data. So having a system where we can add all this data is cool, but what we really needed to implement was a way that people can log in. You can now log in with your own credentials from your own educational institute, and this gives us information about who is this user, but also just have access to your own data, and in the future share that with others that you want to share it with. So implementing a system that handles all this access management is now in there, and really helps us keeping data secure. We also are working on APIs. This is kind of the back end to make sure that people can send us data and can retrieve data from us. So for the tech-savvy people, work in progress. And what you've seen so far is a lot of different services, platforms that ARISE is building. We have the target list, we have ADA, we have the DNA dashboard, we have the AI portal, the sensor portal. And what's coming year is actually one gateway, one portal to rule them all, where you can find all the information that you need when you want to join ARISE, onboard, help. I don't know how to find the button to press the start analysis, or log in and access all the data that you have. So this is what we're working on. I think this was the overview of what the team's been doing. And I think I hand back to Elaine before you have your lunch break. Thanks.
So here we are, after 4 and a bit years. We really hope that in September, when we're really marking our end of 5th year, we go live. We don't know exactly what that means, because it feels like we're already alive. There's tons of researchers already connected. We're connecting with international programs. We're having hundreds of experts helping us to find the reference specimens that we need. But we really want to create this moment where we're really opening ARISE, doing research right now, as we speak, with the sensors, with your own traps that need to be sorted and DNA sequenced, and bring everything together under such login so we can enter the data and also send it to the larger repositories. It will be for researchers as we start, and those associated with research. But of course, we're also venturing into what else can we do for society? What can we do for all of those field managers that really are dying to use this? So work internationally also with other programs to get things going, like biodiversity meets data. I hope you're not underwhelmed, actually, but a bit overwhelmed. And there's lots food for thought. Help us to get to that next stage, and also help us to get really into that operational phase. This is ARISE as we are.